Okay, well, welcome everybody. I'd just like to acknowledge the traditional owners of all the lands on which we meet, and Southeast Landcare would like to acknowledge that the Aboriginal and Torres Strait Islander peoples are the traditional custodians of the land on which we work and play. I'm calling in from Wiradjuri and Ngunnawal country today, and I invite you to type into the chat your own First Nations community if you're able to, and that will just give everyone a little practice of writing into the chat as well. So if you are new to Zoom, you'll find the chat button along the bottom of your screen on the black bar there. So if you click onto the chat and you type in the name of the your local First Nations community, that would be great. Thank you. So as well as paying respects to the elders, past and present of those nations, I'd also like to extend that respect to other Indigenous Australians who are present today. And I'd also like to speak Landcare's commitment to using traditional cultural values of collaboration, inclusiveness and shared responsibility in all our projects and all the work that we do. So my name's Linda Kavanagh. I'm the Southeast Regional Landcare Coordinator, and I'm also the local coordinator for Boorowa Community Landcare Group. I've been involved with Alex James, who is the Southeast Landcare Biodiversity Officer, and also Deborah Butt, who's a project officer with the Yes Area Network. And we've been involved with working together to coordinate this webinar today. So this is one of a series of webinars to promote private land conservation and awareness of biodiversity issues in the northwest of the Southeast region. This is the second webinar. The first one was the Echidna webinar with Professor Frank Gritzner in November last year. And we'll be following up with another one on raptors in the next coming months. So look, watch out for that one. And we hope that these webinars inspire landholders to conserve wildlife and habitats on their land that they manage and provide an opportunity for you to share insights and experiences. This is part of a broader partnership program between New South Wales Biodiversity Conservation Trust and Landcare New South Wales. And the ultimate goal of that program is to build understanding and increase capacity of both the BCT and Landcare groups in our state. So I'm just going to give you a little bit of just a snapshot on how to participate in the session today. We're going to have our speaker talk for approximately 30 minutes. And I'm going to invite you to share any questions that come to you in the chat, and we will be monitoring those questions and then bringing those questions to Josh at the end of the presentation. So during the large number of people on the webinar, I will monitor the chat and I'll ask the questions on your behalf rather than you speaking the questions yourself directly. We will be recording this part of the meeting that Josh is going to be speaking at. So if there's anyone who wasn't able to get to the meeting, or if you'd like to review the session, then you will be sent a link to the recording afterwards. So I'd just like to remind everyone to stay on mute and keep your videos off just during Josh's presentation. And then you are able to put them back on for the Q&A if you want, but it was just might reduce any sort of feedback and interference while Josh is talking. So I'm very, very excited to introduce now Josh Griffiths, who is an ecologist with EnviroDNA. He has been specialising in platypus research and monitoring for the last 15 years. And after many long nights trying to catch the elusive critters, he has overseen the development of an eDNA as a new monitoring technique for platypuses. We will also have Sasha Wells on the call, and we'll hear from Sasha after Josh's presentation. She'll provide some details of the Great Australian Wildlife Search website and that program how people are able to participate in the Murray-Darling Basin Authority citizen science work that's being released. So we'll hear from Sasha later. So I'll hand it over to Josh now, and he will share his screen. And I just invite you to, just keep things going in the chat, and we'll see you on the other side. Thanks, Josh.
No worries. Thank you. Hopefully everyone is able to see my PowerPoint screen now. So, so my name's Josh. I'm, I guess most people call me a platypus ecologist these days. And as was mentioned, I've spent, most of the last couple of decades chasing these little critters around Australia, trying to understand how they're doing, essentially, whether they need to be listed as threatened or vulnerable, what are their sort of habitat preferences, trying to advise people about how to manage waterways for them. So I'm going to share a bit of the knowledge from my research, but also sort of broader stuff about platypus itself. And then also talk about this amazing project that happened almost a couple of years ago now, which at that stage was called the Great Australian Platypus Search. That's now morphed into the Wildlife Search, and talk to you a little bit about the results from that and sort of what's happening next. So, platypuses are pretty adaptable little critters. They occur all along the east coast of Australia in a range of different habitats. This is what I would call is a fairly typical good habitat for platypuses. And the kind of things that they need in their habitat is really, obviously as a semi-aquatic creature, water is the primary thing that they require. And not just having water available, but also having the correct flow regimes and also decent water quality that's going to support their macroinvertebrates. Also, the vegetation that's around waterways is really important to the health of the waterway itself. They hold the banks together to stop all that erosion. They deposit, or they shade the water so that you don't get temperature fluctuations. They deposit woody debris and organic matter, which is all food and habitat for all the bugs that essentially support everything else that lives in those waterways. Particularly for platypus, stable banks is really important, and that's related to that riparian vegetation. So they need those banks to be able to construct their burrows. So when they're not active in the water, they're going to be resting or reproducing or taking shelter in banks. So having nice stable banks, fairly vertical profile so they can construct those burrows is one of the critical things. And we're actually just completed some research that demonstrates how important that is. And then in the water itself, complex habitat is really important. So having woody debris, having this nice sort of cobbled substrate on the bottom. Again, this is providing the nooks and crannies for all those macroinvertebrates, which is what platypus require. And essentially most of these habitat requirements are really about supporting lots and lots of bugs. So really, platypus can tolerate a range of different conditions, but what they do need is lots of food resources in their area. What is less easy to define is how important these different variables are and what happens when they get affected. So how much impact does it have if we clear that riparian vegetation, or if the banks are eroding, and so on. So given that we know this is what they like in their habitat, it's pretty easy to then work out, well, what are the major threats that they face? And essentially anything that's going to impact those key criteria can potentially have an impact on platypuses. And again, it's really difficult for us to understand exactly how severe some of these threats are, but we've got a pretty good understanding of what they are. So obviously things like drought and water extraction, anything that we do to reduce the amount of water that's available in the environment, reduces the amount of habitat that's available for platypus, reduces the amount of habitat that's available for their food resources. It changes the water quality. It can get quite stagnant and warm, and the dissolved oxygen gets really low. It can reduce connectivity along waterways. So these nice healthy rivers become a series of ponds at certain times of the year. And that's not great to support anything really, particularly something that's really mobile like platypuses. We tend to divert water for our own uses. So it becomes a very precious resource that we require. And when we do that, not only are we removing water from the system, we're changing those flow regimes. And also these massive structures are barriers for platypuses moving up and down waterways. So they create, they fragment populations, which is a real issue for wildlife in general. When we clear that vegetation, it leads to erosion of banks. It's more evaporation of water, changes water chemistry. So, this kind of result is an incredibly unhealthy platypus habitat. Native vegetation is so much better than our invasive weeds. So willows in particular are a real problem. Not only are they sort of a monoculture which doesn't support much, but they form these really dense root mats which just don't support many invertebrates, and also makes it really hard for platypus to forage. And then when we have unrestricted stock access, that sort of exacerbates these problems as well. You get more bank erosion, you can get pathogens in the water as well. So these are the kind of major threats that they face on that landscape scale. Other things that they face at a more localized area are things like introduced predators. If they've got healthy habitat, predators aren't really a problem for platypus, but as soon as those waterways dry up and they've got to move across land, things like dogs and foxes can take them quite easily, or if they've got to move around these barriers that we put in their way. I do a lot of work in the Melbourne region, and unfortunately we see a lot of issues with litter. Platypuses are incredibly good at getting tangled in anything that's an enclosed loop, and I've seen some pretty horrific fatalities as well as some pretty bad injuries from things that are as simple as rubber bands or hair ties, really things that are part of our everyday life that we don't really think about, and they can become a lethal object when they get into our waterway. And then finally, these things, thankfully, these don't seem to be much of a problem anymore. We managed to get these completely banned in Victoria a couple of years ago, these opera house nets. Prior to that, they were killing hundreds of platypuses a year across Australia. I now believe New South Wales, Tassie and South Australia have followed suit. I think it's just Queensland that's holding out a little bit now. Since these things got banned in Victoria in 2019, we have not recorded a single platypus drowning in them. So a really simple, easy thing that we can do to just remove one little threat that they face. So the reason that we don't have a great understanding of what's going on with platypuses and the severity of some of those threats is that they're just a really difficult species to monitor in the wild. So, and this applies to a lot of wildlife, but I think platypus are an extreme case. They're very widespread. They tend to be pretty sparsely distributed. They're also highly mobile. They live in an aquatic system, which is a bit more difficult. And they're also just difficult to capture. And they're also mostly active at night. So you can't simply go out and look for platypuses. Typically what we do is we tend to set these nets and try and trap them. So our traditional techniques are very time and labor intensive, which means they're really expensive. I can't put these nets in deep water, so we're already limited on where we're able to actually collect this data. Any of these sort of approaches cause stress and disturbance to the animals, even though we try to minimize it as much as possible. And I guess importantly, they can be really they can really lack sensitivity. So I certainly know that when platypus are in low abundance, I can go out and trap a waterway and I will not necessarily catch any there. So you've got to do repeat surveys to have any confidence that you're detecting platypuses. And ultimately what this means is that we don't have as much data as we want. We either have false negatives, so we sort of don't manage our waterways appropriately because we don't know platypuses are there, or we don't understand what impact our management actions are happening, and also some of those threats that are going on as well. So if we don't understand things properly, how do we address them effectively? So this is just a really quick time lapse of what it takes to actually set these nets. So I tend to set these nets in about 5 or 6 sites along a waterway, spaced about 5 or 10 kilometres apart. They take about half an hour to set up. So it's an all afternoon thing to get these nets set up. I've then got to check them all throughout the night, every couple of hours, so to minimise disturbance to them, and then pack them up at dawn. So it's an incredibly long and labour intensive and cold and wet nights quite frequently. And as I said, probably a third of my surveys, I won't catch anything or no platypus anyway. So it's a lot of work, and you know, even with unlimited budgets, we simply can't do it as widely or as frequently as we might want. So that's why we invested pretty heavily in looking at this technique called environmental DNA. It's something that's really exploded in the last probably 10 years now. And it's really based on the premise that every organism is shedding DNA into its environment. And we've all seen a CSI type program where the detectives go and they swab the crime scene to try to identify the criminal. Essentially, we're using the exact same techniques in a wildlife space. Now, the advantages of this is that we don't have to capture, we don't have to observe the animals, means that we're not disturbing them, it's non-invasive, so that's a huge tick. And I guess from a monitoring perspective, the fact that it can be really cost effective, particularly when doing things at a really large scale. The sampling methodology is really simple, so you don't need specialists out there all the time, you know, collecting these samples. And importantly, it's incredibly sensitive. So a couple of years ago, we worked with a mob at Melbourne Uni to model this. And I'm not going to go too much into this, but essentially we can take just a couple of samples and run a couple of tests on that in the lab, and we get above 95% detection rate. So if platypuses are there, even if they're at really low abundance, we've got a really high probability of detecting them. Conversely, if we don't detect them, we've got a high confidence that they're not there. That's really important for any survey technique. Because it's really simple to go out and take water samples, we're less restricted by environmental conditions or weather, so we can do it virtually anywhere, which means we can do things over large scales. But importantly, instead of relying on anecdotal data like sightings, we can actually design our programs to do systematic surveys and address questions directly. So I'm not going to get too much into the technical side of what eDNA does and what happens in the lab, but just a very brief overview of what happens when a sample comes into us. We extract all of the DNA from that sample, and that includes DNA from a variety of different species, not just platypus and fish, but things like bacteria and fungi and plants and all kinds of stuff in there. And what we've got to do is find that tiny little bit of platypus DNA that's hidden amongst all this other stuff. So we can design a probe, a genetic probe that matches a short sequence of DNA that is unique to platypuses. So it's a genetic code that doesn't occur in any other species. And when we add it to that sample and it goes through a few sort of genetic wizardry in the lab, essentially that probe will bind to its matching DNA, lets off this little fluorescent signal, which is this bit here, and that's what we're able to measure. So depending on the strength of that signal, we can determine how much DNA is in that sample. Now, Victoria probably has the most comprehensive data on platypuses anywhere, and that's because there's been some very long-term monitoring programs, particularly in Melbourne and out here in the Wimmera region. This is just sort of broken up into small subcatchments, which is basically defining some good sort of spatial areas that are useful for quantifying platypuses. And what we know in this area is, so these subcatchments, the red ones, are ones that platypus have either disappeared from or are in some serious trouble. We know that they disappeared from the Lower Murray back in the sort of 60s and 70s, but some of these other ones are a lot more recent, generally within the last 20 years. The orange ones are where we know platypus still occur, but we've been able to see that there's declines or they're pretty sparse throughout those areas. And then, of course, we also have catchments where they're doing quite well. Now, outside of those areas, we simply have very little data to understand what's going on. And this data, this little bit of data we've got here is what sort of resulted in the state of Victoria classifying platypuses as vulnerable a couple of years ago. But it's based on some pretty limited data. And I guess the question that I often get is, well, how severe are these declines? And you can already see these sort of red and orange catchments tend to be clustered in this sort of drier, more modified western part of Victoria. So it's largely agricultural areas. And then, you know, our healthy populations are in that sort of outskirts of Melbourne, where it's in those sort of the Yarra Ranges area. And a lot of this eastern part of Victoria is pretty undisturbed forested areas. So we might assume that platypuses are doing quite well out here and perhaps under threat over this side of Victoria, but we simply don't know. So we've just got these massive gaps in our knowledge to really understand what's going on with platypuses at a state level. And this is where we've got the best data of any of the states. And this is what led to the Great Australian Platypus Search a couple of years ago, was really to be able to systematically look across Victoria and importantly fill in these gaps so that we can understand what's going on. You know, do we actually need to upgrade their status in Victoria? Or on the flip side, are they doing quite well and we can actually take them off that threatened species list? So I was going to have a really quick pause there and maybe just see if there's any burning questions that we want to address before we launch into the next little bit. Linda, is there anything in the
No, I have got something in the chat. Can you hear me?
Yes, I can.
Sorry, I have got, can you identify individual animals or is it as a species level, at the species level when you?
Yeah, good question. It's at a species level at this stage. I would say watch this space in 5 or 10 years time, but the reason for that is that we're dealing with very fragmented DNA, so small pieces of DNA rather than entire genomes. If we could get entire genomes, we could identify individuals, which I think would be amazing. I would love to do a mark recapture study on platypus just using water samples, but at this stage it's just at that species level.
Okay, thank you. And what other survey methods do you use?
Well, it's pretty much live trapping, and eDNA is really about it. People have tried to do visual surveys, sort of systematic visual surveys, but essentially it just has such low sensitivity. Like if you see a platypus, great, you can tell that they're there. But if you don't see them, what's your confidence that they're not there? And because they're mostly active at night, they spend 3/4 of their time active at night. The chance of you actually encountering them out in the day is greatly reduced. It's certainly not unusual to see them during the day, but as a survey technique, that sensitivity that I talked about, which is great for eDNA, is just incredibly low for visual surveys. And so they tend to be okay where you get really high abundance of platypuses, but that's not really telling you much. Usually it's where they're struggling in low abundance, which is where you really want some information. And unfortunately, they're just not sensitive enough to do anything in that space. So it's really just the two methods that can be used.
Okay, and we've got a question here about citizen science and how you can get involved in collecting water samples for eDNA testing. Do you want to answer that now, Josh?
I will get to that in a minute. Yep.
Okay, all right. So there's just one other question at the moment, then we'll carry that one over, is how do you process your data?
I'm not sure.
That was Alex. I don't know whether Alex can maybe...
I mean, essentially what we get at a very basic level, it's not quite as black and white of this, but essentially from every site that we get samples from, we get are platypus there or are they not? There is grey area in there, but essentially that's what we work on. And if you design your program properly, you can look at that in different, say, habitat types or different environmental variables, so that you can look at, what are the conditions that support lots of platypus versus those that support very few. And that's really what the Great Australian Platypus Search was about. So I hope, but it really depends on what your question is for the individual surveys as to how that data gets processed.
Okay, we might follow that one up later, Alex. Thank you. A question from Michael. Is there a best practice website toolbox for bush carers to assist platypus habitat? We know the basics, remove rubbish and plant riparian species. Is there anything else?
Yeah, so there's a little bit of info. I've got some information on the back of our, we created a website a little while ago called Platypus Spot that has some info on it. I know the Australian Platypus Conservancy has some data on, some information on their website as well. So they're probably the two of the key ones. What I would say is platypus conservation really requires some fairly large scale impacts. That they're very mobile, and because they're quite dispersed, you require large areas to support a population. So it doesn't mean that localized stuff doesn't make a difference, but to have a really good impact, it needs to be integrated with a sort of a catchment or sub-catchment scale works to sort of all work together.
Great, thanks, Josh. I might just go to this one more, and then you can get on with your talk, and we can have some more questions at the end. What sort of numbers constitute an abundance of platypus? Like to know.
Short answer is that we simply don't know.
Okay.
Anyone that tries to tell you how many platypus there are, even at a local scale, is completely guessing. Someone tried to do an estimate of how many platypus were in Australia recently, and I think it was something between 50,000 and 350,000. There's just so much uncertainty. We've got no reliable way of estimating numbers, which is what makes the species really difficult to then look at declines across time if you can't estimate numbers. So it's really challenging. And there's ways you can get around that and sort of infer abundance, but essentially we don't know raw numbers at all.
All right. Well, thanks, Josh. I might just throw back to you to, yeah.
All right. And we can, get back to some
Q&As later.
Yep.
Thank you.
Okay. So the Great Australian Platypus Search. So this was really exciting to be able to do. It's something that I've wanted to do probably after my first couple of years of looking at platypuses, realizing that this is required, but we simply haven't had the tools to do it before. So the evolution of eDNA is now provided this tool that we can use. And I guess importantly, so this map in the bottom is our rough outline of what our survey design looked like. So there was about 2,000 sites distributed across Victoria. And importantly, these aren't just randomly thrown out there. These are selected in sites that, one, provide good coverage across their range, but also incorporate a lot of different variables that we want to look at. So with any wildlife survey, it's no point just going out, well, look, these are the areas that we know platypus are. Let's go look for them there, because if you already know they're there, then what's the point of saying that they're there, because you're just wasting your money. But also it gives you incredibly biased results. So if I was just to go to areas that I thought were really suitable for platypus, I'd get high numbers of positive results back. And therefore my conclusion would be, well, platypus are everywhere, they're doing really well. So it's really important to have this randomized approach to your survey design, where you're not just looking at good habitat, but also poor habitat and everything in between as well. So you've got that comparison to say, well, these are the sort of habitat conditions that are good for platypus. These are the ones that are poor. And you can then use that to direct what conservation efforts are important and where to implement them. The other thing I will say about this is that this program was, even though platypus was the sort of flagship species for this program, it was actually about aquatic biodiversity in general. So we looked at general vertebrates throughout all of these water samples, not just platypuses. So even in areas where we didn't expect to find platypuses, it might be really important habitat for growling grass frogs. So yeah, so it was very much about broader aquatic biodiversity. From this, so we had 1,800 sites that we looked at. We also had a couple of hundred audit sites that I'll chat to a little bit in a while. So again, looking at Victoria, so this is the grand total of platypus data that we had in Victoria. And I think this was for 30 years before our survey. So 30 years is the time frame that we tend to look at for conservation status for platypuses. It's about 3 generations. If we filter that a bit more to just the last 10 years, it's incredibly sparse, and a lot of this data is anecdotal sightings, so they tend to be clustered around population centres or popular fishing areas and so on. So it gives you quite a biased information. There's a lot of gaps here that we simply don't have any data, and we don't know whether that means platypus aren't there or no one's looking. On top of that, we had done a number of eDNA projects. A lot of these are little localized projects, so they don't all fit together, but it certainly fills in some of these gaps. Green is where we find platypuses, and the grey symbols are where we don't. So it sort of fills in a bit of the gaps, but it's still pretty sparse across the state. And off the top of my head, I think there was roughly around 1,500 data points that we had in 10 years prior to the Great Australian Platypus Search. And from that, we're able to sort of look at these kind of catchments and go, you know, green catchments are where we expect them to be quite widespread, and our yellow catchments are where they're quite restricted, and then our grey catchments are where we don't expect to find platypuses. And then we could overlay our survey design for this, and hopefully you can see it's kind of filling in a lot of those areas where we simply didn't have data before. So really well distributed right across Victoria, but also keeping into mind, so the other thing we had to think about was logistics. So where can people actually access waterways? So we had to restrict our sites to where there were road crossings or campsites or tracks and things like that as well. So from there, we developed a website. So people that wanted to get involved could log onto this website. They could go onto that map and sort of select sites that were near their house or near a favourite holiday spot and register and say, hey, look, I'm going to do these 3 sites in this area. We would then send them out sampling kits and instructions and send them off their way. And this is just a few of ours. So we had everything from primary school kids to octogenarians to Landcare members. We had an incredibly willing and enthusiastic response from people. And we encouraged them to sort of take photos while they're out in the field so that we could share them and encourage others as well. And so the sampling process was really simple. So essentially you take a water sample with this big syringe, these filters screw on the end, and then you just have to force that water through, and all the good stuff, all the DNA and all the cells gets caught in this filter. That then got a little bit of preservative added to it to help stop DNA degradation. And then that just got posted back to us and we could do all the processing. We developed this app so that people could record all the data, so where they were sampling from, site codes, volumes. And then we also had people doing a habitat assessment out in the field. And this wasn't necessarily for us to look at the sort of conditions that were out there in the data. It was really more of a community engagement exercise and an education exercise to get people thinking about all the different elements that contribute to waterway health. And then we got inundated, so 2,000 sites, that was 4,000 samples that came through our door. And so it kept our lab team pretty busy. We did have some challenges with the app not working and some people recording data the wrong way, but we certainly learnt a lot from that. So it created a few headaches for us in the office. Some of the other challenges was, this was at the end of 2021, we thought COVID was kind of heading out the door. And of course, Melbourne went into a 3-month lockdown. So a lot of the people registered for this program and then couldn't leave their house, which was a bit of a problem. It also made it really challenging for us to get any sort of lab consumables. And when we're either posting stuff out or getting samples back, we had a hell of a lot of problems with delivery delays. And unfortunately, it meant some of our samples got destroyed because the DNA degrades. But we got most of them back, which was pretty good. And then also, partly because a lot of the sampling got delayed until later in the year, was that some of the sites that people were sent to were either dry or they couldn't access them, et cetera, et cetera. So we did have backup sites and instructions for what people could do in that space. But all up, we were aiming for 1,800 sites. We ended up getting data back for over 1,600, which included, we had some sites that we just had to exclude because either the sample quality was really poor or they got degraded because of delays, or simply it was really low sampling volume. But generally, you know, we got an amazing response. I think out of all the sites that were sampled, 95% were done by citizen scientists. So it was pretty amazing. The other question that came up from some of our stakeholders was, well, you know, we're sending people out there with no experience about how to do this. Can we actually rely on the data that we get back? And so that was where these audit sites came in. So we'd flagged 200 sites that some of our team, so professional ecologists, were going to go out and do the sampling at those same sites. We ended up getting to 164 of those. Now, of course, these weren't done at the exact same time as our volunteers because we didn't know exactly when they were going out, but the average difference was 30 days between that. So that introduces a little bit of variation in results. What was quite noticeable was that the volume that we're able to filter by professionals was much higher than what we got from our citizen scientists. And that's not surprising given that, you know, there were 7- and 8-year-old kids and 80-year-old people doing some of this sampling. But even despite of that, we had 82% agreement between our results for both of those. The ones that did differ, there were 12 sites where the citizen scientists were positive for platypus and we were negative. And on the flip side, there was 18 sites that we had positive results and the citizen scientists had negative. So no real bias in any direction. We've probably got a little bit better quality and a little bit better detection rates with the professionals, but generally the information that we got back, the samples that we got back from our citizen scientists were really good and you know, provided some useful information. Okay, let's get on to the results, which is the exciting bit. So if you remember, this is the data that we had on platypuses in that sort of 10-year period before the GAPS survey. This is what we're able to do in just 3 or 4 months. So these 1,600 sites, green is where we detected platypuses, grey is where we didn't detect them. And then our yellow sites are what we call possible detections. It's trace amounts of DNA that quite likely says that platypus are there, but they can come from things like contamination and some other sources. So to sort of reduce false positives, we say that, look, that they might be there, but we're not completely convinced. And I guess when you're looking at these survey results, it's important to look at not just at a site level, but what's going in around them. So for example, you know, there's a little yellow result here surrounded by all these green ones. So you'd almost certainly say that platypus occur there. Whereas this one over here that has a lot of non-detections around it is likely going to be a false positive there. So, but generally the overall pattern is very much what we expected about platypuses. So we get lots of green over here in eastern Victoria, and then in our sort of western and northern reaches, unfortunately, we get a lot fewer. And that agrees exactly with what we've seen in our more systematic or more detailed surveys. And then, of course, you have some healthy clusters, for example, down here in the Otways, which again is what we expected. Now, I will say there were some really interesting results that popped up. We had a detection down here in southwestern Victoria in a catchment that platypuses hadn't been recorded in, I think, over 50 years. Here in the Wimmera, we actually know platypuses are there. There's a little remnant population in this one waterway here, but this is the furthest downstream that they've been recorded in, I think, 20 years or so, prior to the millennium drought anyway. So that was really exciting. Some other anomalies down here on Wilson's Prom. Platypuses have never been detected down there. And I will say that was a really faint trace. It was almost on the borderline of being a possible detection. And another unusual one here, which is out near where I am at the moment, out here in East Gippsland. So what we did, we sort of looked at where these unusual detections were and sent some of our team out there to look in a little bit more detail. Oh, and then I will say there was also some areas where we thought we might detect platypus a bit more where we didn't. So a couple of these catchment areas where we know platypuses occur or we think platypuses occur, and we still got detections, but just not as widespread as we thought. When we drill down into these catchment areas a bit more, a lot of it is because of this randomized design that we used. A lot of these sites here aren't actually on the main river channel. So a lot of them are on these little tributaries that aren't necessarily suitable for platypuses. And so again, we looked at some of these areas and filled in some of the gaps as well. So, so our diamond markers here are what we then followed up with. And for a lot of these areas, we started seeing a few more detections showing up where we thought, these unusual detections that we, for example, this one here, we couldn't actually find any data to support that. Same down here on Wilson's Prom. And so most likely those are going to be a false detection, but it'd certainly be interesting to look at that down the track as well. So just to give a really broad overview, I mean, this was by far the largest platypus survey that's ever done, both in terms of the number of sites, but also the spatial scale. And that's simply because we've now got tools to be able to do that, and also because of our amazing citizen scientists. Importantly, it wasn't a biased thing. It was a very rigorous design and filled in a lot of these gaps where we simply had no data on platypuses. A lot of them are saying that we don't have data because platypuses aren't there, but that's really important for us to know and important for waterway managers to know. The overall pattern really agrees with what we expected. I think our detection rates were a little bit lower than I would have liked, and there's a couple of reasons for that, both from sample quality, but also the timing of the surveys. But generally, it's sort of reflected with what we expected, and it's really highlighted that while platypuses are still pretty widespread in Victoria, there are certainly some issues and some areas where they're quite restricted, particularly in this sort of western and northern parts of Victoria. And so overall, it really kind of supports the fact that, yes, platypus should be listed as vulnerable, and that's probably the right level at this stage. But importantly, what it now provides us is a really systematic and rigorous baseline that we can now measure those changes over time, so we can really identify declines at a lot finer scale and with a lot more rigor than we've ever been able to do previously. So that was what we're able to achieve in Victoria a couple of years ago now. The next stage of this is, like I said, it's morphed now into the Great Australian Wildlife Search. And we're now looking at later on this year to do something quite similar throughout the Murray-Darling Basin. And you can sort of, these are the initial sites that have been identified. There's a couple of thousand there. That's going to be refined a bit further, and there won't be quite as many sites, but you can sort of get an idea of the scale and the scope of it. It's a fairly ambitious undertaking, and this is where we're hoping people can start getting involved. So again, we really want to get citizen scientists involved, local Landcare groups in their local area to get involved and be able to go out and collect these samples. So finally, lots of people involved in the initial project, both from an on-ground thing, from a funding point of view, from an organisational point of view. It's certainly not something that we do in isolation. So huge thank you to everyone that was involved. And oops, I think that's it. My voice is getting a bit croaky. I better stop, I think.
All right. Thank you so much, Josh. And I will just acknowledge that Josh hasn't been feeling the best. So we do thank him for his time today and for speaking for such a long time. I don't know about the others, but I've certainly been inspired about the logistics of the gathering and what you've been able to achieve in Victoria. Really inspired by the number of sites that you've picked up in such a short time. That's sort of, the number of dots that emerged there in 3 or 4 months, and the diversity of the people involved. That's really exciting to see. So so thank you for sharing that story, and hopefully we can now get a bit more information about how we can help fill in those gaps and how we can do similar sorts of things in New South Wales. So I just got a couple of questions in the chat, and then we'll spend some time on the citizen science stuff too. Sasha's just sent a message to say she's not able to join us, Josh. So
okay.
yeah, do you want to sort of deal with that first, or go to the other questions, or what do you think?
No, well, I guess basically what Sasha would have been telling people is that, look, we really want people to get involved and sign up. I think she's just going through the process of getting a website and a registration process finalised, and I think she was going to circulate that to the group in another week or two. So yeah, keep an eye on your emails and you'll get a bit more information coming. And then, as I mentioned, we're working with a number of groups throughout the Murray-Darling area to refine those sampling sites and work out which ones are actually going to be feasible, which ones are our priority ones. And yeah, so that will all get finalised as well shortly.
Okay, just the information that Sasha sent through said that they will be calling for citizen scientists to get involved this spring in 2023. The program has more than 2,000 sites available for citizen scientists to get involved and collect data, and with more sites coming up for autumn and spring sampling as well. And she just wanted to thank the sponsors and funding through the Murray-Darling Basin Authority, and they've got a limited number of free sites, up to 420, or sites available for sponsoring by community groups or individuals at $400. So Sasha will make those details available to this group following the webinar. So hopefully that will, there is more information coming, but she did say the other day when we spoke to her that it was going to be a couple of weeks. So hopefully that will all become clear. So back to the other questions, Josh.
Excuse me, Linda.
Oh, sorry, Alex.
Sorry. Yes, could you just, Josh, would you mind just stopping your screen share, please?
Oh, sure. Yep.
Oh, great. Thank you. Yeah, what's been the impact of large-scale fires in catchments, Josh?
Yeah, really good question. Previously, fires had really thought of a non-issue for platypuses, but obviously the recent ones were just at a scale and severity that no one's ever seen before. Luckily, and one of the reasons why we haven't been able to, well, one of the reasons why fires have been thought to not be a problem is that we never have pre-fire data to compare against, and also it tends to be quite localized and based on trapping. Luckily, we had a PhD student that was doing some pretty widespread eDNA stuff through eastern Victoria and southern New South Wales. The fires actually burnt through half of her project, which was a real problem, but it meant that after the fires, we were able to go back to those sites and resample them and see whether platypuses were still there. The good news was that they were largely still pretty widespread through those burnt areas. But when we actually looked at our control versus burnt areas, there was a bit of a decline there, and it was related to both fire severity and also rainfall events after fire. So generally they got off pretty lightly, but there certainly was a small impact on platypuses, which, you know, is to be expected.
Okay, we've got the next question is from Andrew is, what is the range of eDNA? Do populations upstream show in surveys further downstream?
Yep, so one of the strengths of eDNA in waterways is that it does travel, so you can pick it up over a larger distance. Typically, and the distance that travels varies a lot depending on the river conditions, depending on the amount of DNA, the rate of degradation, all these kinds of things. Typically, in most of our sort of lowland rivers, it's anything from 100 meters up to a kilometre. And given that platypuses move that distance in a night, it's a bit irrelevant really, but yeah, it means that you're not, when you're getting a positive at a site, it doesn't mean that platypus are at that exact point. It means that they're upstream, typically within a few hundred meters.
Thank you. Now we've got a comment here from Erin that ACT are running the Platy project for citizen scientists based on visual surveys. Given the low sensitivity of visual surveys, do you consider this program to be worthwhile?
Depends on what your objective is. I would say, so from memory, I think the way that they're running it is that they're having people out at sort of 10 or 12 sites along a waterway and then sort of recording how many of those sites you see platypuses at. And I think they are repeating it several times. So it's a reasonable design, and I think it'll be good to say that platypus is there, but its capacity to detect change over time, unless it's catastrophic change, like yes, they were there and now they're not, is going to be incredibly limited. And I think if you went and saw a statistician, they would be able to define that for you.
So on the map of New South Wales, Josh, it looked like the coastal areas around Sydney and Wollongong were not going to be monitored.
Yeah, that's what I understand. We were trying to get New South Wales government involved, like the Victorian government did. Apparently they haven't come to the party. So at the moment, the funding is from the Murray-Darling Basin, and so it has to be focused in that area. I would say that I do actually have a reasonable amount of platypus data along that east coast, because I did do some post-bushfire monitoring at about 500 sites along there. But yeah, at the moment it's focused on the Murray-Darling Basin.
So given that a lot of these people here are from the southeast corner of New South Wales, if they wanted to be involved with eDNA surveying, do you think there's an option for us?
I think depending on exactly where everyone is, I think the sites did overlap, so that sort of Yass-Goulburn region, but they don't go all the way over to the coast. As I mentioned, that's not the final design, so I would just hold out until you see the info that gets sent by Sasha. But yeah, unfortunately, because of where the funding comes from, you're sort of restricted on where you can go to. That doesn't mean that if we can find other funding sources, particularly if there's a local industry that wants to sponsor a local area, we can do another dozen sites in that sort of coastal area as well. So it's not set in stone. It's just this is where we're forced to focus on because of where the funding comes from at the moment.
Sure. There's a few people there in Pambula, near Bombala, who are all very keen to be involved and happy to link up with each other as well. So that's great. Ruth Jenkins has a comment there. I believe there is platypus in the Macquarie River at Albion Park, Calderwood. Just put in a submission to council to do. Yes, so a submission to council to see what they want to do with those waterways. Do you have any comment about that area or?
No, I'm not overly familiar with it. I believe that we've had some data from the Macquarie River previously. So yes, I mean, platypus are certainly in the Macquarie River. Don't have much information about how widespread or their status or what's happening with that river, unfortunately.
Sure. Okay, so a few logistical questions. Does the temperature of the water affect the DNA sampling?
Yes, it does. So temperature is one of the key things that leads to DNA degradation. So similar to the distance that the DNA travels, the time that it persists in a waterway is reliant on a lot of different conditions. UV, temperature, and oxygen are some of the key things that degrade DNA. So if you've got warmer temperature, your DNA is not going to persist for as long, and it's not going to travel as far either. But it's going to be a pretty minimal impact on overall detections, I would say.
Okay, and do the floods affect platypus after the El Nino?
Yes, that was one of the things that we did notice, and particularly some of the post-bushfire stuff I was doing in 2022. We actually found detections decreased across the board, and this is where having a good survey design was important. So we found our post-bushfire monitoring had really low platypus detections, and so we could have gone, oh, well, bushfires had a significant impact on platypuses, but because we also looked at control areas as well, and we saw the same decline, it means that effect was across different areas, both burnt and unburnt. And we think we've got pretty good information that it's likely to be because of those floods. Part of that is going to be that flooding is going to displace animals from their waterway, but I think it also affects your detectability. So if you've got more water around but the same amount of DNA, you have a dilution effect. And so typically I recommend people avoid high waters both from a safety aspect, but also from an eDNA sampling, and tend to focus on that sort of low to normal flow range.
Right, thank you. We've got Michael who's worried about asking a silly question, but I'm going to ask a sillier one first. So
There's no silly question, it's just silly answers that I can give.
Absolutely, I agree. So I noticed you say platypuses. So that is
Yes.
that is the proper plural of the platypus. Is that correct?
Yes, most people love platypi for some
Yep.
reason. So because the root of the word is Greek instead of Latin, if it was Latin, platypi would be correct. Because it's Greek, the correct term is actually platypodes, which I quite like, but I don't think I'm ever getting that across the line. So the anglicised version of that is platypuses.
I love it. Platypodes it is. Thank you. So Michael's question was, do platypus ever swim into saltwater tidal creeks or go into oceans?
Yes, absolutely. So I've caught platypuses in estuarine areas. We don't know whether they will survive there permanently. I believe that they probably just come down from fresh water, do some foraging. Those estuarine areas can be incredibly productive. So they forage a bit there, but they've also been known to go out of rivers and into the ocean and then go down the coast as a dispersal mechanism, because they can cross land, but they're pretty uncoordinated and they're very vulnerable to predators. So the way that they disperse between river catchments is to actually go out in the ocean and back up into the next estuary. I think that's very rare, but it has been recorded multiple occasions previously.
Oh, that's great. Now, Erin's just asking if we were to lobby for sponsorship for an LGA or a catchment, are we able to get a ballpark figure on the amount of funding we would be looking for?
Well, it really depends on the size of the area and the sort of intensity that you want to go for. That's really a question for Sasha, but essentially, I mean, you can scale things up and down because essentially it's a per site cost, and roughly it's around $400 per site for so that platypus and wildlife in general. And so we can just kind of, if you can get $1,000, we can do a few sites. If it's $10,000, we can do a catchment area. So it's really how long's a piece of string, but yeah, hopefully Sasha will be able to provide some extra information about what that looks like and yeah, how to go about contacting her about that.
Okay, but about $400 a site is the ballpark.
I believe that's the figure that they've settled on.
Okay.
And
now here's a question from Jason. To what extent does sediment, for example, caused by carp, affect platypus happiness?
Yeah, really, really good question. And we know platypus disappeared from the Murray-Darling, that sort of lower part of the Murray, back in the 60s, which coincides around when carp were introduced in that area. It's a bit of a chicken and an egg situation as to, you know, because the habitat was also degrading at that time, as well as the carp being introduced, which then further degraded things. I would say that I find platypus in a lot of areas where I also find carp, same with trout as well. So I don't think that they have a huge impact. I think it's more the habitat issues that then carp exploit that impact platypuses more than the carp themselves. But I don't have a lot of great data to back that up.
Okay, great, thank you. Now, Peg has got her DNA kits now, and she's on the Pambula River near the border where the river is quite narrow. The river is flowing and it's very cold. And she's asking, should she take her samples now?
I'm curious as to where those kits came from.
Well, Peg, would you like to answer that? Do you want to
unmute?
Unmute yourself, and yeah, we've got time to divert.
Hi, it's Peg here.
Hey, Peg.
I'm by way of default part of the eDNA team now because of the work that I do with birds of prey. So having had some of the founder of eDNA here and knowing him quite well, yeah, we were just interested to see what might be in the waterways here, knowing that there's actually a platypus reservoir up at Bombala. So yeah, we were just
Bombala
interested.
intrigues me because Bombala is sort of held up there as this great place for platypuses, and you can certainly see them quite well at that reserve there. But I look at the waterways around there and I find them incredibly degraded. So I'd be really interested to work out just how widespread they are, or whether that's just a little remnant population there in Bombala that's easily visible. So it's one of those biases around observations of platypuses that I've mentioned. But yeah, so Peg, I would wait until the broader program has been implemented. I know that the idea was, certainly previously, we tried to get everyone to take samples in a short period. I think we initially tried to get it to be a month, typically in spring when most things are more active. Because of COVID, that had to get extended out. But if we can get all of our samples in a pretty short time period, it minimises any variability that you see because of temporal differences and seasonal differences. So yeah, I would hold off until the program's kind of underway and we've defined a period to collect samples.
All right.
I know you're keen. I know you really want to see what's in there, but we'll get there.
Thanks, Josh. Barbara is just saying, this week we found a small dead platypus on the causeway at Foxground, New South Wales. It had about 20 ticks on its body. Would they have caused its demise?
No, so these are actually a platypus specific tick. Can't remember the name of them at the moment, but I find them on virtually every animal I catch, ranging from a couple to several hundred. If animals are sick, you can get high tick abundance, but almost certainly wouldn't have been the cause of death. If it's on the causeway, it sounds like it's probably it's probably a juvenile. This time of the year, juveniles tend to be dispersing a bit. The adult males particularly chase the younger males out of their habitat, and so we get them turning up in really weird places, crossing land, and unfortunately they get run over, they dehydrate, they get hyperthermia. Or they get taken by a predator. Often when they're taken by a predator, particularly a dog, dogs tend to grab platypus. They'll end up with a few puncture wounds in the fur that you can't see. They shake them and they get massive internal bleeding and the dogs disappear and the platypus looks fine, but it's not doing well. So if I was to make a guess, that's probably what's led to its demise.
Right. That's very sad to hear. Now, Peg has just put into the chat there that Erin and her are thinking they could work on some funding around the Bombala area. If anyone was interested to connect after this call, and I'm sure that would be, if everyone's okay with that, then we can put people in touch with each other, or you can write your own contact details in the chat now. Thank you, Peg and Erin. So that's
The other thing I'll mention is if we're looking at new areas, is that we can use the sort of survey design that's being used broadly. So we can chat to La Trobe Uni and just say, well, look, can you apply that same sampling design program in this catchment area? So then it all sort of matches up nicely. And there'll be capacity to sort of vary that a little bit, but it gives you a good basis as to what a survey design would look like.
Oh, that's great. Thank you. And Alex has just put some handy websites in the chat as well. We've got the platypus.asn.au. Rivers of Carbon have a great guide to what platypus need to thrive. And then there's the Odonata link as well on how to get involved with the Great Australian Wildlife Search. So thank you, Alex, for those.
I would say Rivers of Carbon has an excellent guide for landowners for sort of riparian restoration in general, and also some guidelines about how you can sort of assess your land and say, well, what is it missing? What does it need? So they've got an excellent website.
Yeah, that's great. They definitely do. Now we've got a comment from Barbara. Said she's also got echidnas on her property and they've had ticks. Does anyone know about echidna specific ticks to monitorings? We'll get Professor Frank back.
If I was going to have a guess, I would say that they're probably a different tick than the platypus have, but probably closely related.
Okay. Right. We've got any other questions? I think we've covered the ones in the chat, but is anyone else wanting to ask Josh something? Or Josh, have you got anything else that you'd like to share with us while people are thinking more questions?
Nothing that immediately comes to mind, but I think my email is going to get circulated as well. So if anyone has any burning questions or wants to talk about a platypus monitoring program in their area, feel free to reach out. And yeah, if I don't get back to you quickly, do send me a reminder, because I get a bit distracted. But yeah, do feel free to reach out and get some advice.
Okay, Alex is just saying that she can share the Echidna webinar link as well. So those that missed the Echidna talk will be able to hear from Professor Frank. So we will distribute that as well. And as I mentioned right at the beginning, to the next in this sort of series of webinars, we're planning to do one on raptors. So that'll be coming in the next few months. So just watch out for that space. Is there any other burning questions, or would anyone like to ask anything else of Josh? We're a little bit ahead of time. We've got some time. Oh, Alex has just put the YouTube link in the chat. So Alex, that's the Echidna webinar, is it? Yeah, thank you. All right.
I should say that even though sort of next stage is looking at the Murray-Darling Basin, I mean, the idea is that we do want to do this in all states. I know Tassie has shown some interest, and Queensland as well, and then eventually sort of moving over into South Australia and WA as well. So there are sort of plans to basically make it an Australia-wide thing, which of course we have to move away from platypus a bit, but yeah, the actual biodiversity data we get is quite amazing. Looking all things from fish, frogs, turtles, even sort of stuff that lives around waterways. We pick up a lot of birds and mammals that live in those riparian zones as well. So it's quite amazing data that comes out of it.
It is fantastic, isn't it? So Ruth's just saying that she saw a platypus in Tassie once in the middle of the day. Is that sort of unusual? Because you hear that it's often
Not at all.
Dawn and dusk, or
yeah, I mean, you most likely see them at dawn and dusk, particularly dawn. I find those first couple of hours after dawn. Certainly not unusual to see them out in the middle of the day. They're an incredibly adaptable little critter, but yeah, they do spend 2/3 to 3/4 of their active time at night, which is why they're difficult to see in a lot of places.
Okay, we've got Deborah's just saying, once the data is collected for the next stage, what's the next step in the vision?
Yeah, well, the vision is to expand it across Australia. And then, I mean, I think what the plan is that all this data, I mean, we are just collecting the data at this stage, but the plan is to make that available for everyone. It's got to go through a few QC processes, but the idea is that it will go on to something like an Atlas of Living Australia or the state biodiversity one, so that it's accessible to everyone. Researchers can do their own analysis of it. But I think the important thing is it's that baseline that then we can detect change over time. So whether it's done annually or every 5 years, one of the problems I have with platypus is that, so we can now collect really good data currently about where they are and get an idea of, I'll say, abundance by looking at how many sites are occupied. But I've got very limited historical data to compare it to, so I simply can't quantify declines effectively. Even though we know that they've occurred, I can't provide the hard data to say how much and how widespread. So now we have a rigorous baseline that we can go back in a year's time and go, okay, well, it's similar to what it was, or it's declined by 10%, or it's even increased in these areas where we're implementing management actions. So that's the kind of objectives of this data.
And would platypus across Australia be a variable species, or are they sort of localised? I'm not quite sure. That was from Michael, whether in
So are there sort of different subspecies? I think you're getting at there, Michael. They are all the one species. There is a bit of genetic variability across their range, particularly in sort of far northern Queensland and Tassie. There's a little bit of genetic variability there. One thing they do is they vary a lot in size, and this is pretty common for mammals on a latitudinal gradient. So you get really small platypus up in northern Queensland. Adult males are about a kilo and 30, 35 centimetres long. In Tassie, where it's a lot colder, the waterways are very cold, I can tell you. I think I've still got the record down there of 66 centimetres and 3.7 kilos of a male. Look like a small furry crocodile. And it's purely just a body size can retain heat better. So in colder environments, you tend to get larger body size compared to those warmer environments, but they are all still the one species.
And is there a photo chart of river conditions available?
Well, so that habitat assessment that we were sending around and getting people to do, I added photos in there. So essentially there's 6 different variables you look at, and then within each of those, they all get categorized by, I think there's 5 different scales from really poor to really good. I added some photos in there to try to illustrate them from people, but that's nowhere near an official thing. I'm happy to share that PDF around for people if they find it useful, but that's the only one that I'm aware of.
Okay, and are there any current projects or plans to reintroduce platypus into prime habitats?
Well, a reintroduction of platypus has just happened in Royal National Park outside of Sydney. The question is that a prime habitat? I think there's certainly some question marks around it, or even whether they were there previously is a question mark. There isn't, and the reason being is that generally reintroductions do not work. Most reintroductions fail, which unfortunately we never hear about. And that's simply because the reason that animals have disappeared from an environment is that it's no longer suitable for them. And so putting animals back in there, even though they might persist for a little while, eventually they're going to die out because the habitat is no longer suitable. So unless we can be really clear that we have got rid of whatever those major threats were and returned all of the habitat variables that they require back to a suitable level, it's really, really difficult to get animals to persist again. And also because platypuses are so widespread, they're difficult to breed in captivity. It's never going to be an effective conservation status for platypuses in particular. It generally doesn't work for most species at all, even though zoos love captive breeding programs. But essentially it's very expensive. It's very often not successful, and our money is much better spent in protecting their habitat rather than trying to reintroduce them.
Do they breed well in captivity, Josh? Do you know?
Not at all. So for over 50 years, there was only one animal that was ever bred in captivity. They actually used to die quite often in captivity within their first week. I think it was like 80% mortality that used to happen. Now our zoos are set up a lot better and our knowledge is a lot better. So at Hillsville Sanctuary and Taronga, they seem to be able to breed them fairly regularly now. But when I say regularly, that's sort of, one young a year. And then, of course, you don't have the capacity to house them for very long. So platypus require these fairly large, elaborate tanks. You can't hold hundreds of animals at the one time, which you need to do reintroduction programs. So even though we can do it in terms of a conservation strategy, it simply won't work, unfortunately.
Okay, and are eDNA samples more accurate for platypus as they are aquatic compared to other Australian mammals?
So not so much accurate, I would say. So it's more effective, simply because in aquatic systems, you're able to filter large volumes of water and the DNA disperses and distributes throughout a waterway. The same theory applies in terrestrial systems, and the lab side of it, the sensitivity is still the same. the challenge you've got is trying to find the DNA in an environment. So terrestrial animals are going to leave DNA where they come in contact with things, and then the DNA stays there. And you can't filter large volumes of soil to extract a little bit of DNA. So it's a lot more challenging to find the DNA in other environments. We certainly do stuff. We find bat DNA in the soil in caves, so you can be quite targeted about where you sample. But eDNA works a lot more effectively in aquatic systems, and we can't see stuff in the water. We can't set cameras for water for aquatic animals like we can in terrestrial systems. So yeah, it's certainly a lot more useful for aquatic animals.
Thank you. We've got a few people who've had to sadly leave now and run off to something else, but Alex has just put in the chat if everyone is happy to be contacted about the next webinar. This database will get used to distribute the details for that future webinar, if that's all okay with everybody. And yeah, look, we've got a few more moments if anyone else has got something else for Josh. Josh is happy to stay on for a bit longer if you've got some more questions. Otherwise, we might wrap it up early. No? Okay, all right. That sounds like you've just been a wonderful presenter. Thank you so much, Josh. And we look forward to getting that information from Sasha about the citizen science project, and we'll distribute that to this participant list so that you'll get that with the recording to the series. So hopefully, or recording to this session. So hopefully you've enjoyed hearing from Josh, and we thank you very much for your time today. And thank you for your support of South East Landcare and the Northwest webinar series. So
no worries at all. Thanks everyone for listening to me ramble on.